Welcome to this very important presentation on antibiotics and their effects on the microbiome. Other than diet, there is no other factor which plays such an enormous role in determining our microbiome. In fact, one could argue that it's more significant. Either way, almost all the talk over the years surrounding the use and overuse of antibiotics has centered around drug resistance. And yes, I'm not discounting that. However, I am one of the few voices screaming from the rooftops that we are exterminating our microbiomes. And this comes with grave consequences. An antibiotic-resistant pathogen affects a tiny fraction of us. However, the dramatic disruption in our microbiome from antibiotics affects most of us. I know that generally people want to run out and get a prescription for any ailment, especially for their children, but some 30% of these are unnecessary, and it could be argued more than that. We're always so concerned about things in the short term, but don't consider the long-term repercussions. Fear tends to outweigh logic, but consider this presentation and at least you'll be armed with more information than anyone else you can bump into. Antibiotics were invented in the 1930s, and their benefits have been fantastic. They have been one of a handful of factors which have contributed to a significant improvement in health and longevity over the past almost 100 years. I'm not disputing that. However, there were people who lived well into their 90s before the advent of antibiotics. They were just fewer in number. Before 1900, the world was quite different. In the United States in 1900, the average life expectancy was about 47 years old. If you survived childbirthing, both mother and child, disease, famine, accidents, and war, then you might have just done well for yourself. I suppose they just felt lucky and accepted the deaths of those around them as an unfortunate part of life. These days, to die at less than 70-something seems semi-tragic. We have 401ks, plans to pay off mortgages, and maybe some traveling in our so-called golden years. Point is that we don't accept death for a variety of reasons, and thus we are all determined to do whatever seems best to guarantee our well-being. And given this, your microbiome pays the price due to the semi-neurotic use of antibiotics. We have evolved with this foreign ecosystem inside of us for many millions of years, and we should take care of it before we alter it permanently for the worse. In addition, what is supposedly the next most important thing after your health? Well, in today's world, I'd argue it's money. Antibiotic use in agricultural animal production goes back many decades. When it was first discovered that their use in animals significantly increased growth and feed efficiency, their use exploded. If you're a business, you can grow bigger animals for market off of less feed, and then you get to put more money in your pocket. This has been proven over and over, although the exact mechanism is still speculated. It's probably a blend of a few things like decreased satiety hormones, lack of competition for nutrients, and decreased gut volume. You see, the gut, ours included, requires a tremendous amount of our energy, some roughly 30% of your reserves, just to maintain itself. The short-chain fatty acids and other signals produced within the healthy gut help feed cellular turnover, mucus production, and other things that require a lot of building blocks. If you eliminate or reduce these good actors, then you may be left with a less healthy animal which is able to put on weight more efficiently. Sound familiar? Disease and obesity are far too common in the United States and other so-called developed countries. Maybe it's time you think outside the box. Although antibiotics are even used more in animals than in humans, we still have quite the selection. From this comprehensive meta-analysis, we see here in Table 1A the effects of antibiotics on aerobic bacteria. As a rule, but certainly not always the case, aerobic bacteria have more bad actors, more opportunistic pathogens, while the true health promoters are very oxygen sensitive. So here we see a list of common bad guys, such as Enterococcus, Escherichia, Klebsiella, Staphylococcus, and Streptococcus, names I mention frequently. The color red indicates an increase in their presence. Within each cell are the references they highlight. As you can see, there's a lot of red. But wait, these are bad guys. Shouldn't they be reduced by the use of antibiotics? Well, we'll see more of the specifics of that later. Just note that, for example, in the penicillins, a lot of data indicates amoxicillin increases bad actors across the board, while the cephalosporins as a whole increase the genus Enterococcus, which houses some very bad actors. And this continues to the macrolides, tetracyclines, and so forth. That's quite the list. We'll highlight a handful of the most popular ones in this presentation. On the flip side, we see here the anaerobic bacteria. Again, as a rule, these are health promoters, like Bifidobacterium, Coprococcus, Eubacterium, Faecalibacterium, Roseburia, and Ruminococcus, names with which you're becoming familiar if you watch my presentations. In the penicillins, we see a lot of green. That means reductions. In the cephalosporins, we see more green than red. And the red we do see, for example in C. diff, is the intended consequence in treating a C. diff infection. However, giant point of note, the number one cause for C. diff infection is antibiotic use. More on that later. Also note for the rest of this table, the significant increases shown for Candida, as in yeast, across all antibiotics. Like bad bacteria, Candida is also an opportunistic pathogen waiting to take advantage of an environment which serves it best. When you wipe out the bacteria to include the good guys, Candida has a free pass to become opportunistic. Returning to the anaerobic bacteria, we see a lot more green reduction here, especially for clindamycin and here for ciprofloxacin. So let's look at some of those good guys who we saw that got impacted from our previous table. In this study, healthy subjects were given a 10-day round of ciprofloxacin or clindamycin or placebo, and their fecal microbiomes were evaluated at 6 time points out to 1 year. We can see in this figure, at various time points with each antibiotic, the true health promoters of the gut were significantly reduced to include Faecalibacterium, Roseburia, Lactospira, Allostipes, Coprococcus, and Ruminococcus. The most profound shifts were in the first month. However, in general, the microbiome inhabitants recovered over time, although at 1 year out, the good guys had generally not returned to their initial levels. This is a common finding, is that the microbiome can generally handle a round of antibiotics once in a while, and generally recover in a month or two. To focus on the bad actors, in this study with compromised patients, the results showed that metronidazole increased what they called Enterococcus domination threefold. What is that? They defined it as when at least 30% of the microbiome was occupied by a single taxon. Now that very rarely happens, and if it happens with a species from Enterococcus, you're in very bad shape. You can see the results in Figure B up to 35 days post-antibiotics. Interestingly, the Enterococcus domination, largely caused by metronidazole, one class of antibiotics, caused a 9-fold increased risk in vancomycin-resistant Enterococcus bacteremia, a different class of antibiotics. If we take antibiotic use to a bit of an extreme, here we have 16 healthy, brave souls who took ciprofloxacin, vancomycin, and metronidazole for 7 days, the goal being to measure their combined effects in the healthy. The colors in the heat map, Figure C, indicate logarithmic changes, black a reduction, white an increase. The green arrows point to health promoters I've established over the years as the former director of medical education for a microbiome firm. You can see massive differences from day 0 to the collection of fecal samples on day 9. Several key health promoters were reduced over 100-fold, while a classic opportunistic pathogen, Streptococcus, was significantly increased, as was the genus Lactobacillus. Again, your probiotic behaving like a bad actor. In a very comprehensive and invasive study involving more healthy controls, these researchers administered a substantial antibiotic regimen and then measured in detail how their microbiomes recovered over 180 days in 3 arms: natural recovery, what they called spontaneous, by fecal microbiome transfer, or with probiotic supplementation. Fecal microbiome transfer was the clear winner, as it always is. More on that later. Natural recovery was in second place, although 3 key health-promoting taxa never fully recovered, which is often seen after antibiotic use. And the use of probiotics in post-antibiotic recovery had the undesirable effects of significantly delaying microbiome recovery until the end of the study and causing a bloom in Enterococcus, which is a bad genus we've already highlighted. I know many people recommend the use of probiotics during antibiotic use. This paper may be reason to rethink that strategy. You can see here in this study the common antibiotic pattern this time by colors. Within the blue rectangle, you can see at 9 days post-antibiotics, drastic changes from day 0 in the colors which represent the abundance of various genera. Most obvious is a huge increase in the color red, which represents the genus Streptococcus, a genus full of bad actors we saw earlier. Also, again, we see a huge increase in Lactobacillus, acting like an opportunistic pathogen once again. That's the stuff in your probiotics, by the way. Lactobacillus may be good for infant and vaginal health, but my data shows the opposite for adults, especially those who are dysbiotic. The authors go on to state, quote, "We observed profound changes shortly after the course of antibiotics, with a drop in microbial diversity, overgrowth of the genera Streptococcus and Lactobacillus, and an early loss of anaerobic bacterial taxa with important roles in short-chain fatty acid metabolism." That last part, anaerobic taxa with roles in short-chain metabolism, those are the health promoters. There are a number of interesting things I could point out from this study, but let's just say that their overall findings were in accordance with everything else. Post-antibiotics, bad guys up, good guys down, and with time, a partial but not complete recovery. But here I want to show you something different. In Figure 5, we see significant increases in the first 2 time points, or what are called virulence factors. These are some of the things that make bad guys bad. These are increases in the expression of their genes, which are involved in iron uptake, bad bacteria love iron for growth, LPS, the coating of their outer membrane, which the immune system is highly reactive to, and genes involved in adhesion and invasion. These are also classical traits of opportunistic pathogens who want to adhere to and invade the cells that line your gut. As I've said a couple times now, after one run of antibiotics, the microbiome generally recovers. The real challenge is when someone has many rounds, especially in quick succession. This is when the real problems can begin, and especially if they have a bad diet and take PPIs. These researchers took only 3 subjects labeled D, E, and F and administered 2 courses of ciprofloxacin, then analyzed the fecal microbiomes. These 2 courses were separated by 6 months, which is still a bit of time. I'm sure some of you had one course after another before, in less than 6 months. As usual, there was a recovery, but it was not complete. Each gray vertical bar represents a course of Cipro. You can see, especially for subject F, that when you look at these 3 health-promoting taxa, Allostipes, Subdoligranulum, and Roseburia, there was a loss of presence in the microbiome, as highlighted by the red circles showing a disappearance in detectability. It's not so much that antibiotics do a terrible job of killing off the bad bacteria, it's that they do a really good job of killing off the good bacteria. As I say, and I think every presentation, at the root of our many diseases is dysbiosis, an imbalance in our microbiota. It's the good guys who keep the bad ones in check. With continuous hits to the microbiome, the environment shifts from one which is health-promoting to one which is disease-promoting. Nowhere is this better exemplified than in C. diff infections. By the way, I'll soon have an entire presentation dedicated to this. This slide is from a C. diff study, looking at the microbiome in hospitalized patients. In general, they noted a large correlation between fluoroquinolones and Enterococcus abundance. And in Figure H, they show huge blooms of Enterococcus, shown in red, in conjunction with vancomycin. We can learn from this that the microbiome is not static. Just look at the fluctuations in these taxa denoted by the color shift. We also learn that all antibiotics are going to affect you in one way or another. Best to take care of yourself. Prevention truly is the best medicine. As we continue our dive into antibiotics and their association with C. diff, we have this paper looking at the effects of vancomycin or amoxicillin versus placebo on the microbiome. Again, the usual suspects went both up and down. You can read that in the text. In the figures, I have something new for you. This is the effect on bile acids. I have a whole presentation dedicated to this complex topic, but let me explain what we have here. You see, the microbiome is able to modify the bile acids, which are produced by your liver and excreted into your duodenum. They are in fact hormones, so their effects are quite powerful. They aren't just for digesting fat, which is what most people will think of first. There are two main types, primary and secondary. As you can see in B, the level of primary bile acids collected in the feces skyrockets on vancomycin, whereas in D, the secondary acids fall off a cliff with vancomycin use. These are both bad. Altering the microbiome by antibiotics use altered the bile acid profile. This combination is the root cause of C. diff. You can view all the deeper science in my presentation on C. diff infections, but let's just do a quick review. C. diff spores are just about everywhere, and they're exceptionally hardy. At some point in any given year, you'll test positive for C. diff when doing a microbiome analysis. If you're healthy, don't worry, it's normal. A C. diff infection is not so much an infection when it's a normal inhabitant of the gut. It's a problem when the checks and balances are broken. With antibiotics use, as we've now established, we killed off the good guys and the bad guys thrive. One of the things that these good guys do is metabolize bile acids. The first step is simple hydrolysis. About 1/4 of your species can do this. The second step is dehydroxylation, to convert primary bile acids to secondary. About 0.0001% of your microbiome possesses the gene to make this complex enzyme. This balance between primary and secondary creates an environment to enable germination and growth of C. diff or prevent it. And this balance is determined by your microbiome, which is in turn determined by a few factors, in this case, antibiotics being of paramount importance. To continue with bile acids, and these finished children, macrolide use was associated with a 10-fold increase in the troublesome genus Agarthella. And as you can see in figure D, as time passed, the genes which direct the production of bile salt hydrolase took many months to recuperate. Again, from our previous slide, BSH, bile salt hydrolase, was the first step in converting primary to secondary bile acids. So, even though some 25% of your microbiome possesses this gene, this too takes a hit from antibiotics use. The authors go on to state that it took 2 years for some aspects of the microbiome to recover after just one single round of this antibiotic. So perhaps now you understand the statement highlighted from this paper, which says in one sentence what I've been saying for a few slides. And although antibiotics are the #1 culprit, PPIs do a lot of damage to the microbiome as well, and are an established risk factor for a C. diff infection. For more on PPIs, watch my presentation entitled Stomach Acid Blockers and Their Effects on the Microbiome. I hope you can begin to understand how and what we do impacts those little critters inside of us, with whom we're ideally supposed to live with in harmony. C. diff is just a classic severe case of dysbiosis. But the same players who are involved in C. diff are the same opportunistic pathogens involved in many other conditions. And who are these actors? Here's a chart of mine after a meta-analysis of the microbiome in various facets of C. diff. I used 30 human fecal microbiome studies in total. The blue box lists a range of cohorts. This is the microbial signature of C. diff. The orange color indicates an unhealthy association, while green indicates the opposite. Enterococcus, Escherichia, Veillonella, and once again your probiotic Lactobacillus are all highly correlated with C. difficile, in one way or another. Whereas in the healthier cohorts from these 30 studies, many of the amazing health-promoting taxa I've identified over the years were significantly higher. These include one name with which you're probably familiar, Bifidobacterium, which is the other main genus in your probiotics. But other names you've likely not heard of before, such as Allostipes, Eubacterium hallei, Roseburia, Ruminococcus, and Faecalibacterium prausnitzii. When these good actors in green are significantly reduced, they allow the opportunistic pathogens in orange to become opportunistic. Let's look at this from another angle. The standard treatment for both initial and recurring C. diff infections is antibiotics. Yes, more of the stuff that got us into this mess to begin with. Crazy, right? That's modern medical thinking for you. Kill, 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 instead of nourish, nourish, nourish. In this review of therapies to treat C. diff, when FMT, fecal microbiota transplant was done. In every case, it performed better than antibiotics. The column is entitled Most Cost-Effective Treatment, which means most successful. What is FMT? It's when you wipe out the microbiome of recipient with a potent bolus round of antibiotics and take feces from a certified donor to inoculate the patient. It sounds disgusting, but it's highly effective and without a doubt shows the power of the microbiome as evidenced here and in many other studies. Now, considering that most of our disease/conditions are correlated to dysbiosis in our microbiome, C. diff being the most severe instance of it, and if FMT is able to help these people so dramatically, then don't you think improving the microbiome can help others? Well, it does. I've done it. Not with FMT, that's reserved for extreme cases, at least for now. But we can modify your microbiome with the next best thing, driving significant environmental shift with the intelligent use of properly blended and dosed prebiotics. The purpose of this presentation is not to contradict the orders of your practitioner, but to inform you. Anyone in the healthcare community with half a brain knows that antibiotics are overprescribed, and with that come the long-term risks that everyone is now familiar with, antibiotic resistance. But the definition of overprescribed is different for different people, and herein lies the problem. For example, I was able to dramatically help a girl of 20 years of age who had estimated that her antibiotic exposure in her brief life was about 100 rounds. This may seem excessive to you, and it is, but to her parents, it was in their mind the right thing to do. And I am willing to bet your line in the sand is higher than mine. So this presentation is both an opportunity to reconsider future prescriptions for questionable concerns, and also helps you to reflect on the past. Oftentimes, one or more rounds of antibiotics in succession are the trigger to someone's disease. So think back. If you suffer from a condition, what happened in the weeks and months before the symptoms started? Were there one or more rounds of antibiotics? If so, the antibiotic-induced dysbiosis in your gut could very well be the root cause. Let's fix that. If you found this presentation informative and you think addressing the health of your microbiome might be a good idea, then in the Protocols tab of my website, themicrobiomeexpert.com, you can find a science-based protocol for this condition, among many other conditions. You're also invited to view and read my testimonials in its respective tab.